Okay, so last thing I wanted to go over primary, secondary, and tertiary articles. So here, I'll just break down some of the quick stuff right now. So with a primary, you're going to notice some of the first things is that the title's very specific. It almost sounds like a hypothesis. There will be a lot of different authors, and there'll be this bold text right here, and that's called an abstract. Then you have strict guidelines for the next few sections, such as introduction, and it's all background information. You'll start to see methods and then results. And this is the key. This is the key that separates primary from everything else. And it's images like this. You see how there are like significant values right there, error bars, like actual graphical data. So like raw data, graphs, measuring everything. This is what should give away a primary. Because review articles like secondary, sometimes they'll have images, but they're more like illustrations. So these are all graphs. They're actually making measurements. They're saying that one is higher than the other, for in this case, a gene knockout in this case. And then finally, you have a strict discussion section at the end of primaries, as well as just essentially just mountains of data, observable things, the word significant, and then just the language that is written in is usually very strict and full of lots of thuslies and such. So a secondary source usually has a more generalized title. In this case, this gives it away, just pathogenesis of human lymphomas. It's just going to be a big general article, usually a lot fewer authors. So there still might be an abstract, so watch out for that. But essentially, you get into an introduction section, but then it just goes off into different sections. Origins, signaling, this kind of signaling. And you do see figures, but if you can tell, these are kind of more they're generalized. They're kind of more like illustrations than they are data. If you don't see any significant error bars, things like that, maybe some percentages are fine, but realistically, you're gonna kind of see overview images, and that's what you're seeing here. And they're usually just a lot longer. So if you can see here, like this is just a diagram of a signaling gene. This is a diagram of an actual gene. It's not something that, they're making any conclusions off of. They're summarizing conclusions, and they're also huge usually and have way more references than the primary. Tertiary is usually the easiest one. For example, right here, World Herald staff. These are non-scientific articles by science journalists or just normal journalists, and they're usually just expanding on things that people care about. They're translating essentially the findings, and in this case, it's a medical finding. New cancer therapy clinical trial at UNMC, good results after other treatments, and it's written in the language of just how a normal person would want to read it. There's usually like a more informative images like this that will appeal to broad readers. And let's see. Essentially, tertiaries are the easiest because you can tell right from the source who's writing it, and if it's a newspaper or a magazine or something, it's tertiary. And then if you want to take a closer look, just read the language. If it's more colorful, it's definitely a tertiary.